Our final module is on genes and exercise performance. There'll just be the one lecture this week and some small readings to give you some time to finalise preparation for the last test. It's been known for hundreds of years that many biological traits are inherited, whether it's in plants, animals, or indeed humans. The identification of DNA as the carrier of genetic material in the early 50s really then led to an explosion in our understanding of genetics, molecular biology, and greater insights into the link between genetics and disease. And that, of course, has extended into investigation of the links between genes and exercise performance. And the question, are athletes born or are they made? The early approaches in this field looked at comparisons of twins, maximal oxygen uptake, and indeed some of the later studies looking at muscle fibre type. You can see in this early study comparing the VO2 max values of twin A and twin B for monozygotic twins, a very close association, with a little more scatter with the dizygotic twins, suggesting a large degree of heritability of this VO2 max. And in fact, in this study, they suggested that the heritability could be as high as 80 to 90%. In a more recent study, the Heritage Family Study, a multi-centred trial, a really classic study in exercise science that brought together exercise physiologists and geneticists to really look at the relationship between families, genes, and some exercise-related variables. If you looked at the pre-training, the sedentary VO2 max values, there was a familial aggregation of VO2 max, and you can see here a heritability estimate of about 51%. So clearly there is a large genetic component to many of the physiological variables that influence performance. And of course, one of the major determinants of athletic performance is sex, and we know that that's very much determined by the genes that you inherit and from your parents. There's been interest in, can we identify genes or even polymorphisms within genes that might predict or help us predict success in certain types of exercise performance. One of the early candidates was the ACE gene, or the angiotensin converting enzyme gene. And this, if you think about the biology of angiotensin, renin, it's involved with cardiac and cardiovascular physiology. And this study showed a relationship between the various alleles in this gene and performance, at least measured in climbers. And you can see that the more athletic climbing group had a greater frequency of the II genotype. And this was associated with an ability to increase performance in relation to an intervention. The other gene that's received the most attention, and it's perhaps the one that is most successfully linked with measures of athletic performance, is the alpha-actinin 3 gene. Now, alpha-actinin 3 is a protein that's involved in the cytoskeleton, if you like, of skeletal muscle, and it really is a gene for speed. That's what it's been termed. And you can see in this study from the Australian Institute of Sport athlete group, the XX characteristic, which was, or the combination of the XX, which was associated with an alpha-actinin deficiency, you can see that there tended to be more of that in the endurance type athletes and none of that combination in the power or sprint athletes. And so it's possible that perhaps the alpha-actinin 3 gene might be involved in some way. The challenge with all of these is the ability to predict the athletic potential from a single gene test. And I think, as we all suspect, exercise performance is very much a polygenic characteristic determined by not just multiple interactions of multiple genes, but also multiple interactions between genes and environmental influences. And we'll come back to that point in a moment. One way to look further is to try and see if there are collections of genes that might predict power and endurance. And here, obtaining a total genotype score from the frequency of various genes which might collectively contribute to, say, sprint performance or endurance performance. So in this case, you can see a collection of genes that have been associated with power-oriented sports: the ACE gene, the alpha-actinin 3 gene, angiotensinogen, myostatin, which is a growth-related factor, interleukin 6, nitric oxide synthase. And you can see that, yes, while the power athletes had a significantly higher genotype score, you can see that there was significant overlap. And again, reinforcing the notion of the polygenic nature of performance together with the interaction of the environment. Similar thing for endurance type sport, this time focusing on some of the genes that have been shown to be involved in mitochondrial biogenesis. And as we saw in the endurance performance lecture, there is a close association between muscle oxidative capacity and endurance performance, so that it's likely to think that something related to mitochondrial biogenesis or genes related to mitochondrial biogenesis might have some association with performance. And here we have nuclear respiratory factor 2, PPAR alpha, PPAR delta, and PGC1 alpha involved in mitochondrial biogenesis. Again, the genotype score, and yes, the endurance athletes had a significantly higher value, but again, a great deal of overlap. Another aspect is there a genetic basis to the adaptations to exercise? So not only the intrinsic value, if you like, but in terms of response to training. And here again, we go to the Heritage study, and they showed that in response to a standard 16-week exercise intervention, there was quite a degree of variability in the response to that standardized training program. When they looked at, from a genome-wide association study, a number of polymorphisms that were associated with the trainability, and connected those polymorphisms together and grouped them with those that had fewer than 9 and greater than 19, you can see that the more of those genes that you had, the greater was the VO2 training response. So clearly there are many genes that are likely to contribute to this response, but the fact that there are some, and there seems to be this relationship, suggests some degree of genetic basis to the variability in the VO2 max response to training that was observed. So here is a figure from a very good reviewer or article looking at the interactions between genetics and environment in determining athletic performance. And you can see, there are many factors which are going to influence performance, which are determined by nature, if you like, the genetic basis. Sex, height, VO2 max trainability, skeletal muscle characteristics, and other relevant traits are going to have heritability indices which vary. And some of these you can see based on some of the literature, the proportion that might be attributed to some of these polymorphisms. In parallel with that, you can see many of the factors which are influence, or which would influence perhaps the expression of that genotype. So early exposure to physical activity, early exposure to certain environments, for example, altitude. And so in relation to some of the debates around the success of the East African athletes, is that a genotype factor, or is it related to early exposure to regular physical activity, to altitude exposure? There may be many other things that contribute to that. Coaching, training techniques, access to facilities, and all of these things are going to work together. And so the champion is going to come from the pool of elite athletes, which in turn is going to be influenced by the variable combinations of the genetics and the training environment. And so while I think there's no question that successful athletes need to choose their parents carefully, they need to work hard and take full advantage of all of the opportunities they have to maximize that genetic potential. So are champions born, or are they made? Well, they're probably born and made. Just to finish on a few aspects of exercise and genetics, we know that exercise can affect gene expression. If you remember back to the lectures on the muscle adaptations to exercise, a single bout of exercise can impact on gene expression. And here is from my particular research interests in the regulation of the glucose transporter, the GLUT4 gene in muscle. Here is a single bout of exercise which increases the expression of GLUT4 at the gene level in skeletal muscle, and that continues during the recovery period. And as I showed you in that lecture, a number of factors and signalling processes which can impact on gene expression. So factors related to energy balance, AMP kinase, factors related to calcium, all impact on transcription factors which influence the expression of the GLUT4 gene. One of these is an enzyme that's involved in histone regulation. And this raises the issue of what we call epigenetics, or modifications to gene expression that are not dependent on changes in the nucleotide sequence. And this is where environmental influences potentially can exert themselves. And so it's possible that gene expression can be altered not through any change in the nucleotide sequence, but through changes or modulation of structures and molecules around the gene. So in the case of DNA, it's surrounded by proteins known as histones. And these histones can be modified, they can be methylated, acetylated, phosphorylated. And in fact, the GLUT4 adaptation to exercise involves the phosphorylation of a histone deacetylase enzyme, which results in increased acetylation of the GLUT4 promoter region. So it's possible that exposure to exercise, exposure to various environmental stimuli can modify gene expression, not through altering the nucleotide sequence. And this is how environmental influences might modify gene expression. Some of these will be transient, some of them can be maintained for quite some time. The question then is, can they be passed on to the next generation? And there's a lot of activity in this space at the moment. Another aspect of the regulation of gene expression relates to non-coding pieces of RNA referred to as microRNAs. And it's known that these microRNAs can influence gene expression and the ultimate phenotype. And variation in the levels of these microRNAs might be implicated in some of the adaptive responses, and indeed might alter gene expression and the ultimate phenotype. So here's an example where strength training was examined in a group of healthy volunteers, and just as was seen in the Heritage study with variability in VO2 max, there was a variability in the increase in lean body mass with this strength training program. There were low responders and there were high responders. If you then looked at the microRNA profile of the low and high responders, you could see here that the low responders had increased levels of this microRNA in muscle known as 451, whereas the high responders had very low levels. So perhaps something about having an elevated level of this particular microRNA attenuated the strength training adaptation in that group of subjects. So there's been a lot of interest in these various non-genomic modifications of gene expression and phenotype. Another aspect of exercise and genes is the ability to manipulate gene expression, and in so doing manipulate characteristics of muscle and exercise performance. And there are a number of examples now where single genes have been introduced into muscle to affect performance. So notwithstanding my earlier comments about exercise performance being the result of many factors and polygenic influences, it has been shown in a number of studies that if you increase the muscle expression of certain proteins, in this case PGC1 alpha, activated MEF2, heat shock protein 70, PPAR delta, that's associated with changes in the metabolic and oxidative capacity of the muscle and in endurance performance. So here's just one example where transgenic mice were manipulated and there was an increase in the muscle expression of PGC1 alpha. Now this transcriptional coactivator is involved in the regulation of mitochondrial biogenesis, in angiogenesis, and these transgenic mice, in many respects, take on the characteristics of mice that have been exercise trained. If you compare the muscle oxidative capacity, in this case through measuring cytochrome B, significantly higher muscle oxidative capacity in this transgenic mouse. And when they were asked and put on a treadmill to run, you can see that there was an improved endurance performance in these transgenic mice. These are the sorts of studies that really cause some concerns around the regulatory authorities who are trying to regulate and stamp out doping in sport, because it is technically possible to introduce some of these vectors either directly into muscle or, in this case, systemically. And here's an example that is of research in the muscular dystrophy area, which is designed to try and improve the lives of young boys who suffer from this terrible muscular disease. And this disease arises from the lack of a protein known as dystrophin. It's one of the largest proteins in muscle, and using these adeno-associated viruses, it's possible to introduce a microdystrophin and target it to skeletal muscle. Now you can imagine, and you can see here, that when that was done, the green stain here is the injected animal that normally lacks dystrophin. You can see here in the untreated mouse, but quite good expression both in muscle and in heart of this protein that was normally missing. And of course this raises the spectre of what's been called gene doping, where you might inject various constructs into muscle or systemically to try and alter the physiological metabolic characteristics of tissues with the ultimate aim of trying to improve performance. So as I said, it's these sorts of studies where the introduction of genes into specific tissues or into the body can impact on performance that has led the World Anti-Doping Authority to specifically ban gene doping, just as it's banned the administration of various growth factors and other pharmacological agents such as steroids. So, as I said, there have been remarkable advances in molecular biology since the isolation and characterization of DNA, the Human Genome Project, and these have far-reaching implications for our understanding of genetics and disease, and also our understanding of how the body adapts to exercise. But the dark side of that increase in knowledge is the potential for it to be used illegally in attempts to try and improve sports performance.